everyone, and welcome to today's webinar, Elevate Your Research with Spatial Insights. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, please visit leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speaker, Dr. Quyen Tran, Global Business Excellence Manager, Data and Analysis at Aivia. For a complete biography on our speaker, please see the Presenters tab. Dr. Tran, thank you for joining us. You may now begin your presentation.
Hi everyone, welcome to the webinar. My name is Quyen Tran. I am the Global Business Excellence Manager for the Data and Analysis team here at Leica Microsystems. Today, I have the privilege of talking to you about Aivia 13, our latest release, which will help you elevate your research with spatial insights. What does this mean? Essentially, with Aivia 13, we bring you the tools you need to go from image to insight, particularly in your 2D multiplex images. Going from viewing and interacting with your images, to detecting your cells, to identifying phenotypes within your image, to critically examining your data to generate insights. Here's a brief video to show you some of the tools we will be talking about today. In this release, our team came together with the goal of helping you understand tissue microenvironment through spatial biology. For those who are not working in this field, spatial biology brings together two pieces of information about the tissue microenvironment: cell identity and cell location. Using these two pieces of information, we can determine the different types of cells that are located within the tissue and where they are located in connection to one another, giving us the ability to unravel the complex biological interactions at play here in both normal and abnormal states. In the last 2 decades or so, this field has greatly expanded. In the chart on the right, you can see that the number of publications which include spatial biology has rapidly increased, leading to an expansion of our knowledge of various tissues, as well as the need for tools to process the information we are generating here. To gather this information, scientists have utilized several techniques, including microscopy to examine cell location and protein profile, sequencing to look at the transcriptome and the genomic profile, and mass spectrometry to examine protein profile. If you are a spatial biologist using mass spectrometry, I highly recommend looking at our Aivia plus LMD solution that provides you with the tools you need to identify your cells and extract it for mass spectrometry. However, for today, I would like to focus on a different set of techniques, using microscopy and multiplexing for spatial biology, especially spatial proteomics. With this methodology, we can obtain both the cell location and the cell identity using one system, the microscope. In the array of images you see here on the slide, you can see this methodology can be used for a variety of tissues, from colon to brain, as well as different levels of multiplexing, those with less than 10 markers, all the way up to 30 markers. In Aivia 13, we can help you turn these images into insight. Let's dive in and discuss the Aivia features that will be available to you. First, viewing the images. In Aivia 13, we've implemented some new color palettes here that are especially designed for images with lots of channels to help you view them easier. The ability to zoom and navigate in large 2D multiplex images, and a mini map feature to help you navigate between different regions of the tissue. I do want to make a note here that our free viewer called Aivia Community will also have these capabilities and will allow you to view your images on multiple computers, whether these computers are in your lab, in your office, or even with your collaborator. Furthermore, if you've processed your data in Aivia, you can open them in Aivia Community and view the results there also. Now let's take a look at the tools for the next step in our workflow, detecting cells. In Aivia 13, we are introducing two new recipes, cell analysis cell pose and multiplex cell detection. In both of these, a modified version of cell pose has been integrated into the recipe for accurate nuclei and cell membrane detection. Cell analysis cell pose is optimized for a single cell membrane marker and allows you to detect the cell membrane, the nucleus, and the vesicles within a cell. The multiplex cell detection recipe, on the other hand, is great for images where you have multiple cell membrane markers, and it will provide you with output of the cell membrane and the nucleus. Now, for those who don't know cell pose, here's a quick summary. Cell pose is a generalist deep learning cell segmentation algorithm that was developed by Stringer et al. In 2020. In this work, they utilize a variety of images capturing different cell morphologies, microscopy techniques, and a host of other things to generate an algorithm that can segment a diverse range of cells with high accuracy. A snapshot of this paper is shown here on the right, and I highly recommend exploring this article if you have not done so already. Things may sound a little complicated here, but in Aivia, we have integrated this into our recipe so you can utilize it without in-depth knowledge of deep training. Beyond the two new recipes, we have also improved several of our current tools in order to handle large 2D images. Recipes such as the nuclei count, cell count, and our machine learning powered pixel classifier has been updated. In the images above here, I'm showing a few snapshots of this expanded capabilities with the raw images here at the top and the segmented detected results here at the bottom. For cell detection, we've also implemented multiple ways for you to establish regions of interest to help you speed up your analysis. You can manually create your regions of interest, and with the freehand tool, these regions of interest can be whatever shape you would like for your tissue. Aivia also has ways to automatically detect your tissue, whether it's a single piece of tissue or multiple tissues within your image. This way, you can work on the specific regions that are important to you in the images that you have. Now let's look at the next step in the workflow, identifying phenotypes. With Aivia 13, we are introducing two new tools for this. First, the AI Power Phenotyper. In this tool, it's going to be driven by your expert knowledge of each phenotyping. You can determine which phenotype you want to identify, which markers are important, and the level of expression of each of these markers for each of the protein. The overall workflow for this tool includes three main steps. First, providing examples of your specific phenotype to Aivia. Aivia is going to take those examples and train an AI classifier to identify those cells. And then you can apply this classifier to the full data set or the region of interest you've already dictated. On the right-hand side here is an example of a phenotyper that we've trained to identify 3 different immune cell types: CD3 and CD4 positive cells in yellow, CD3 and CD8 positive cells in pink, and CD20 and CD45 positive cells here in green. The second tool is Aivia's new automatic clustering tool, which is going to allow you to identify cell phenotypes in a much more data-driven manner. In this workflow, you just specify the measurements you wish to include in the phenotyper. This can be marker intensity-based or morphological measurements. You do not need here to specify the combination of markers associated with each phenotype or the level of expression for each of these markers for each of the phenotype, just which measurements you want to use. You also have 2 options of clustering methodologies here: K-means, which is a well-established methodology, or phenograph-laden, which is relatively new but has been shown to be effective with complex large dimensional data. Each method here is going to be loaded with default settings, which you can modify if you wish. And then once applied, Aivia is going to pull all of the measurements that are necessary and then begin to cluster your cells into your phenotypes for you. In the example above here, the cells within this pancreatic tissue was detected and then automatically clustered into either 4 groups or 10 phenotypes here. With the automatic phenotyper, the transition from 4 to 10 clusters required only one parameter change. This technique of automatic clustering does offer some key advantages for you. One, it doesn't require you to have in-depth knowledge of all the phenotypes that are present within your tissue. You can be selective about the markers you want to use, but that next level of knowledge, which marker, which amount of expression is critical for each phenotype, is not required here. 2, it allows you to easily expand and scale up the number of markers you use for the phenotyping. You just simply go back to your setting and select a few more markers. This allows you to use 10, 20, even 30 markers for your phenotyping. Third, this large-scale data-driven method can help you identify phenotypes that may be unknown to you, or those you might not have even considered when you were setting up your experiment initially. Now let's move to the next step in our workflow, generating insights. In Aivia 13, we're introducing some new interactive maps for you here. On the left-hand side, you'll see an example of the dendrogram, which provides you with a look at the relationship between cell clusters or phenotypes with multiple measurements. You can select which measurements are displayed, and it's shown here as a heat map to help you compare across the clusters. On the right-hand side, you see an example of a summary chart, which allows you a more focused view comparing one measurement across your cell clusters and phenotypes. Next, we've added dimensionality reduction tools into Aivia's charts. For those who are unfamiliar, dimensionality reduction is a powerful tool to help you visualize and understand data sets with high numbers of dimension by representing them and allowing you to view them in a lower number of dimension. So for our multiplex data sets with 30 dimensions, this can be represented now into 2 dimensions for easier viewing. In Aivia, you can utilize these tools to help guide you find the right combination of measurements to achieve clear decision boundaries when you're looking and setting up your cell phenotype. Let's take a look at a non-scientific example to help us illustrate this. Here is a popular example of an example data set that contains 70,000 samples of fashion-based items that are represented in 784 dimensions. You can just imagine the complexity of looking at 784 dimension and parsing out and beginning to understand that data. So using UMAP, we can represent this in a 2-dimensional format. Here in the UMAP, we see clear decision boundaries, especially when we examine the cluster on the right, which is well defined and separated from the rest of the data. If we dig into this a little bit more, we see that this right cluster consists of sneakers, ankle boots, and sandals, which are all shoes and different from those in the other fashion items. Now, a more biological example here, we can see there are clear boundaries for the cluster on the left-hand side here. We may deem that this is a sufficient decision boundary and continue to use the set of measurements we use for this phenotyping, or you may say this is insufficient and I want to add more measurements or replace the measurements so that dimensionality, so that the decision boundaries is much clearer perhaps in this region of the UMAP. So dimensionality reduction using UMAP here can help guide you in the decision of which measurements to use to separate and to have those clear decision boundaries for your phenotypes. And if you prefer, in Aivia, you do have the options of two other additional methodologies for dimensionality reduction: PacMap and TISNI. So you don't have to do UMAP, which is the example I provided here. Now, one more chart to introduce to you, which is the bin scatter plot to help you visualize lots of cells better. The chart on the left here is the more traditional scatter plot, where each cell of each phenotype is individually charted with multiple colors. This is usually a great way to examine the data, but because we have such a large number of cells, the degree of overlap here makes it really hard to understand and parse out the data. Therefore, we've created the bin scatter plot, where each cell phenotype is aggregated by bin, and then the size of each of these circles represents the number of cells of that phenotype within a bin. So for example, here we can see that moving from left to right here, we see that there's an overall decrease in the number of green cells and an increase in the orange cells here, right here. Now, once I say that, I think we can take a look back at the traditional scatter plot and see that a little bit for ourselves, but it's really hard to pull that information out, whereas in the bin scatter plot, this information is a little bit easier to see. One last thing to show you before the demo, Aivia's object relational tool, which allows you to establish a link between two different phenotypes and then obtain measurements such as distance to the nearest cell or count all the cells within a specific distance. Additionally, we can take these measurements and color code them in the image for a more visual representation of the data. So in this example I have here on the right, the immune cells are in teal, and a cell phenotype called GLUT1 positive cells here are color coded based on their distance to the immune cell. Those in red and orange are relatively close to an immune cell. Those in yellow and green are much further away. So in this snapshot, we can see overall the spatial relationship and distance between these GLUT1 positive cells and the immune cells here. Now with that, I'd like to transition us into the demo portion of the webinar so you can actually see the tools that I've talked about in action here. As this is a broad audience, I do want to mention just a couple of things here about the data set we'll be working with before we launch into Aivia. The data set that we'll be looking at is a pancreatic ductal adenocarcinoma tissue that's been imaged using Leica Microsystems cell dive system with 30 markers for a broad examination of this tissue. There's one marker I like to call special attention to, which is GLUT1. This is a glucose transporter, and for this data set, it denotes for us the region of the tissue that's experiencing hypoxia and is where aggressive cancer cells are located. which is one of the reasons why in the demo I'm gonna be focusing on this region and looking at the cells that are GLUT1 positive and the relationship and the interaction it has with other cell phenotypes within the image. Now let's move into Aivia. All right, everybody, here we are in Aivia with the pancreatic ductal adenocarcinoma tissue. Let's take a quick look at some of the visualization tools we talked about earlier. Let's change the color palette here to ice. And let's zoom into our image. I'm going to use one of the presets here in Aivia at 10% here. And while this is a beautiful section of the tissue, I'm actually going to take advantage of one of the other new features, the mini map, and relocate us to a different section of the tissue for our next step in the workflow cell detection. And the reason why I like this area is if we look at the GLUT1 expression, remember that GLUT1 for us in this tissue denotes areas of hypoxia and aggressive cancer cells. So I do want to make sure that when we optimize the cell detection, that the cells in this GLUT1 positive area are well detected. This tissue area also has areas of low GLUT1 expression here. And so I think if we optimize the cell detection recipe here in both high and low GLUT1 expression, we'll get a recipe that's going to work well for the rest of the data and allow us to detect the cells in this whole tissue. So let's get to that step. So I'm going to change the layout here for us. If you didn't know, in Aivia, you can have you can set layout presets, and in each of the layout, you can determine which tools are prominently displayed and where they are displayed. So for the cell detection user interface here, I got our image, I got our channels, I have our annotation tab to do some manual measurements. I also have the object tab to take a look at the results, the ROI or region of interest manager, and the recipe console and the pixel classifier. For the detection here in this image, we're going to utilize one of the new recipes, multiplex cell detection, because in this image, we don't have a single cell membrane marker that stains everything. So we'll need to combine a couple of cell membrane markers to detect all of the cells within this tissue. So let's go ahead and select those cell membrane channels here. I'm going to select a few for us so that GLUT1, it's got a really nice cell membrane stain. Some CDs for us, 3, 4, 8, 45, a few more down here. And for your image, feel free to use whatever combination it's necessary for you to detect all of your cells. We definitely want to detect a region of interest here. I'm going to use the draw tool and select an area that's got both that high GLUT1 expression and area of low GLUT1 expression for us. Telling Aivia to utilize the region of interest for the recipe apply. Let's go in and make some quick manual measurements for us here. Let's gather some cell diameter. Let's say that's about 4 as the average minimum nucleus size. All right, that looks to be about 5. And then I'm going to leave everything here with the default channels. In our Aivia wiki, you'll find information that details the description of each of these parameters, what they mean, and tips and tools on how to optimize it for your particular data set. So for this, I'm going to actually collapse the annotation tab so we can see a little bit more. Turn on one of our cell membrane markers so we can check the results here. and let's see the nuclear membrane that's been detected. Overall, looks pretty good. Maybe a few things I want to optimize if we were doing this outside of a demo, but I think overall pretty good, enough for us to move to the next step. Let's check that cell membrane. Again, overall pretty good. Couple of spots I might want to change if we were outside of the demo, but I think overall good enough for us to move to the next step here, which is phenotyping. So let's transition to that layout. Phenotyping. Essentially, we swap the recipe console and the pixel classifier for the object classifier. Let's go ahead and create one here. We're going to do first the automatic phenotyper / classifier with the cell membranes. The measurements here, standard indicates that we're utilizing mean intensity measurements for the channel. If you want to use something else like morphological measurements, you can by selecting the custom measurements here. For today, we're going to use the standard mean intensity measurements. I'm going to select basically all of the channels except DAPI. This is essentially using 29 channels for our phenotyping. And because we're using so many channels with so many dimensions here, I'm going to use the phenograph laden methodology, utilize the default settings here, and click apply. Again, in the Aivia wiki, you'll find additional information detailing what each of these parameters are and tools and tips on how to optimize it for your particular data set. Now, what Aivia is doing is gathering all of the measurements that is needed for the phenotyping and then clustering the data for us. So with this, we've got 4 clusters here. I'm going to go ahead and utilize another new feature here in Aivia 13 called Spotlight, and essentially spotlight these clusters along with color coding them based on the color of each of the clusters. So let's zoom out a little bit here so we can see more of our region of interest. So here we can see that cluster 1 seems to be evenly or somewhat evenly distributed within our region of interest. Cluster 2 seems to be located more in the region where it's got that GLUT1 positive staining. Let me turn that on so you can see it a little bit better. So that high GLUT1 positive staining section here. Cluster 3 seems to be in this area where it's got the low GLUT1 expression, and cluster 4 seems to be throughout the region of interest here too. So utilizing the automatic phenotyper and the spotlight capability, we can cluster or phenotype these cells and also see where they are located within the tissue and how they're spatially related to one another. Now let's move on to the phenotyper. Gotta give it a name here. We're going to use the new phenotyper and also do this on the cell membrane. Setting a couple of things for us to easily select some markers here. All right, so for this phenotyper, I want to make a phenotyper for the immune cells. I'm going to make 2 classes here. Let's have the first class be our CD3 positive and CD4 positive cells. Let's have our second phenotype in class here be CD3 positive and CD8 positive cells. So now we need to provide Aivia with some examples of what this looks like. So let's turn on these 2 channels and find areas to teach Aivia what these phenotypes look like. All right, I think I see a region of double staining here. Excellent. Let's go ahead and select some of these cells as examples. All right, assign this to the correct group, CD3 and CD4. Now let's do the CD3 and CD8. See a couple of double staining here. Let's zoom out, see if we can find just maybe one or two more. Assign these to the correct group. Specify which channel is most important for each of these phenotypes. So this one CD3 and CD8, and then this one CD3, the first one CD3 and CD4, and then this one CD3 and CD8. And let's teach this classifier, and let's apply it to the whole region of interest here. The results are displayed here. Let's go ahead and use that spotlight feature again. Zoom out a little bit. So not a ton of immune cells here in our region of interest. A couple here in this area, and a few CD3, CD8 kind of here and in this area here. Now, one more tool to showcase to you here in this data set. I do want to pull up the spreadsheet and show you how to do the relational tool. Let's add a relationship here. I want to look at the GLUT1, the relationship between the GLUT1 cells and the immune cells. So let's go from the immune cell group and find the nearest GLUT1 expression, which I believe is cluster 2. And create this. You'll see the measurement here, specifically the distance of these objects to one another. However, we can go in and actually change this and color code it to that measurement. Apologies here. Let's pick the right object distance. And here you'll see let's turn on that GLUT1 expression so you see a little bit better. As we move from the areas outside of the GLUT1 expression, these cells are color-coded red, and as we move closer and closer here to that expression, you'll see these immune cells turn that blue color. So we do see a number of immune cells here in this section that has GLUT1 positivity, but I do see that kind of deep into the areas of GLUT1 positivity, we don't see a ton of immune cells here. They seem to be more located in the vicinity of the GLUT1 expression, but not a lot in the interior of these sections. But here you can see that the color code, we can detect we can set the relationship between these two types of cells and calculate the measurements between these two cell populations and phenotypes, and then also color-coded here in the image for an easier way to visualize that relationship. Now I'm going to transition us to actually taking a look at the large, at this same demo data set, except with all of the cells that have been detected. Here we are, full data set with all the cells detected using the multiplex cell detection recipe that we examined earlier. I've also performed the automatic clustering using the phenograph-laden method, creating 20 cell clusters or phenotype. Additionally, I've created and applied an immune cell phenotyper. This phenotyper is different than the one we had before, as it contains one more cell population, CD20 and CD45 positive cells. We weren't able to capture the cell phenotype before with our previous ROI, but with the full data set analyzed here, I was able to add this into our phenotyper. Now, I'm going to guess that some of you, if not all of you, had questions when we were performing the automatic clustering, specifically how you're going to dive into the biology of these clusters more and get a significantly better understanding of what markers and measurements exist within these clusters. Don't worry, that information is accessible. One great way to view this broadly is utilizing one of our new charts, dendrogram. In this dendrogram, we're examining the clusters or phenotype created by the automatic clustering. We're looking at the standard intensity measurements for all 29 channels, since that's what we utilize to generate the cluster. If you set up your automatic clustering with a custom set of measurements, no problem. You can change this to custom and select the measurements you use. Let me go ahead and expand this panel a little bit so we can see the full details of the markers. So here on the left-hand side, you'll see the list of markers and their expression level across the clusters. On the bottom here, you'll see the cluster listed. And in this dendrogram, you can clearly visualize the expression profile of each of these clusters. And within each of these clusters, what are high expressing markers and what are low expressing markers. In this view, you'll also get to compare and contrast the marker expression profiles of each of these clusters to each other. With this new dendrogram chart, you have a global view of the cellular phenotype profiles within your tissue. Now I want to dive into the GLUT1 positive cells, those that denote regions of hypoxia within our tissue. For this, I'm going to transition us to a new plot, the summary view. This chart's going to allow us to essentially zoom into the dendrogram and compare a single measurement across the clusters. So the default measurement here is AE1. Let's change that to our GLUT1 mean intensity. You can see here that you can pull up any measurement that's calculated and generate the chart from there. Here we see one cluster, cluster 4, with the highest level of expression of GLUT1. Let's go and highlight this cluster in our image. Let's zoom in and adjust our view to see this a little bit better. First, let's check to see if the spatial relationship we saw between the high GLUT1 expressing cells and the immune cells within our small region of interest still holds true when we examine the whole tissue slice here. I've emphasized the immune cells so we can see how they're spatially related to one another. The immune cells are now displayed in pink. Most of the immune cells seem to be not located in the interior of the cell aggregates. Let's zoom in a bit to see that clearer. Again, there are still most of the cells near the GLUT1 positive cells, but there's not a ton of them inside these regions, maybe a few here and there. Therefore, it doesn't look like the pancreatic ductal edema in the pancreatic ductal adenocarcinoma tissue. We see a ton of migration of immune cells into the hypoxic areas here. I do have another hypothesis I wish to explore in this data set. Some of you may already be thinking about it too. The hypoxic area here represents a shift in the cell environment and one that requires the cells to adapt in order to survive. We already see one of the ways the cell has adapted with the expression of GLUT1. So I'm curious to know if the cells within the hypoxic area have also perhaps begun to express or overexpress anti-apoptotic marker as another way to survive. So in order to explore that, I'm going to transition us back to the dendrogram and then focus us on the anti-apoptotic markers we have in this panel. Let's see what clusters have high expression of these markers where they are spatially located in connection to the GLUT1 cell. So for the BCLXL, I see that in cluster 15. Let's take a look at where those are located. All right, I see a few pop up here, but not close to the hypoxic region. The same trend holds in this area here. And down below, we see regions of high BCLXL expression, but not within the hypoxic area itself. So let's take a look at the next marker, BCL2. The highest expressing cluster here is cluster 16. Don't see a ton of them here. Let's move a little bit up. Here they are. Let's zoom in. So I see a few here, but again, not really within the interior of these hypoxic zone denoted by our GLUT1 positive cells. Last marker here with the high expression in cluster 17. I see a few pop up here, but again, not a ton within the hypoxic area itself. Here are more of cluster 17. But again, not a ton of them within the actual hypoxic area, just a few. Additionally, if we examine cluster 4, the one with the high expression of GLUT1, we see a relatively low expression of these anti-apoptotic marker. So from this image in our exploration, it seems that the cells directly within the hypoxic area are not surviving based on an expression or an overdrive of anti-apoptotic marker. They're likely utilizing a different mechanism to survive and grow within this environment. There's still so much to explore here with all of the tools in Aivia and with this rich data set of 30 markers. However, we will have to stop here for today. I hope you were able to get a sense of the potential workflow you can utilize to examine your cell phenotype, how they're spatially connected, and to generate insight into your tissue microenvironment. For now, I will transition us back to the presentation and give a few concluding remarks. A quick note to our current Aivia subscribers. You can now update your Aivia to get access to these new features. For those in our Aivia Go package, you can get access to the visualization improvements for large 2D multiplex datasets. You can also access improvements to the nuclei and cell detection, as well as the pixel classifier to allow it to analyze large 2D datasets. You'll also be able to use the automatic ROI detection. For customers who are in our Elevate and our Apex package, you'll not only get access to those features, but also to our 2 new cell detection recipes, our 2 new methods of cell phenotyping, and all of the new charts that we spoke about today. Of course, I can't end this presentation without thanking the entire Aivia team here, from our developers to our PM, apps, and Bex team, to our director, to our sales and customer success team. Thank you very much for all of your hard work making Aivia a success, and for the successful launch of Aivia 13 here today. With that, I would like to thank all of you for attending the webinar here today. I appreciate your time here, and thank you very much.
All right. Well, thank you so much, Dr. Tran, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. We've already got some great questions coming in here. And Dr. Tran, this first question asks, are images with lots of channels or markers required to use the Aivia 13 tools?
So that's a good question. And while I did showcase in our demo presentation an image with 30 markers, it's not necessary for you to have so many markers to utilize the tools. You can use images with as many channels as you want. There's not necessarily a limitation here. So we can do things like 4 channels, 10 channels, 30 channels. The tools are flexible enough for us to handle the images that you are acquiring.
Great, thank you. This next question asks, does Aivia only work with 2D images?
Okay, no problem. So the tools that we have launched in this particular webinar and today in 13 is optimized for large 2D dataset, as I mentioned, but the tools within Aivia are broader. Even in our Go subscription packages, users can use 3D tools, and we have a number of them for 3D cell detection, 3D object detection, and you can use some of the charts and some of the tools that we talked about today in those 3D applications also. So it's not limited to 2D.
Perfect. Great. Thank you. Next question here. How many channels can I use to phenotype?
Good question. So again, the answer to this, much like the first question, is that it's flexible. So here, you saw me use 29 markers. You can use less than that. You can use up to 30 markers. And remember also that not only are you able to use the markers, so the intensity, but you can also use some of the morphological measurements. For example, what's the size, the cell area, what's the cell perimeter, things like that. So you can use quite a number of combination of different measurements really to do the phenotyping.
Great, thank you. Another question for you here. Do you have any speed and accuracy benchmark data?
Yes, we do. We do have some of that, and that information will be available on our product page And you can see that in that information. I won't necessarily go through everything now because there's a lot of that.
Great. All right, we're still getting some great questions in here. We have a few more minutes, so we can definitely take a few more questions. This next one asks, can we work with Mica, Stellaris, and CellDive datasets?
So yes, so these three systems are part of the Leica portfolio. So we have worked with these teams to make sure that these types of data and the images are coming from these specific system do work with Aivia 13. However, I do want to make a note here that you don't necessarily have to utilize just Leica-based solutions and Leica-based images to utilize Aivia. We can read a number of file formats that are outside of that system, and you can pull in those images and use the tools that we talked about today and other tools that are currently available in Aivia too.
Great, thank you, Dr. Tran. And a follow-up question to that, what about Thunder?
Thunder, yes. So Thunder is, again, another part of the Leica portfolio, and that is something that we can utilize into the system too. So Aivia is pretty kind of agnostic in the sense that for Aivia, an image is an image. The acquisition of it is something that Aivia doesn't really kind of factor into much when it comes to the tools.
Great, thank you. All right, next question here. What kind of computer do I need to use these tools? Supercomputer, laptop?
So this is going to vary a little bit in terms of the answer here. And a lot of it is purely based on the size of your image here. So again, you saw me do kind of a small region of interest here in the large image, and you can use a much kind of lower end system, even a laptop. Some gaming laptops will be able to process things like that. Once you get to larger and larger images, while even a gaming laptop is able to process that, it's going to take a bit of time. So the more kind of high power computer that you have, the faster it's going to be. Not necessarily the whether or not you can do it, it's just the speed of which it's going to be accomplished. We do have some minimum and recommended computer specs on our website so that you can take a look at what we recommend in terms of computer systems.
Great, thank you. All right, another question here. Is Aivia Go included in the AI parts that you presented today, or is it an add-on?
So the AI components that we talked about today, such as the pixel classifier, which is utilizing machine learning, is a part of Aivia Go. There are other tools that we didn't get to cover today because it lies a little bit outside of the new features that we talked about. So some of the 3D recipes that are in Aivia Go also have cell pose, and you can utilize those those AI components as part of that package. As we move to the Elevate and to the Apex, you'll get more and more AI tools, but everybody within the Aivia package subscription tiers will have some tools that have AI components.
Great, thank you. All right, next question. I have used k-means in the past, but I was not happy with the result. How is your clustering method different?
So if you've used k-means before and you weren't happy with the result, I suggest to try out the phenograph-laden method that I showcased in the demo. There's a couple of things here that would kind of help you here. One, it has a capacity and it's been showcased to be very effective in data sets that's got lots of dimensions, lots of channels. We saw that in the demo with the 29 channels. And it's also a little bit less sensitive than k-means to outliers. So if you have some outliers in your images, or you anticipate perhaps there are outliers, I would definitely go ahead and do the phenograph laden. Of course, you can always have the option to run both and kind of compare and contrast the results and see what makes sense for your data.
Wonderful. Well, thank you so much, Dr. Tran. We have time for one more question here. And this final question asks and mentions, I'm interested in cells with particular morphological characteristics. Can you use phenotyping or your phenotyping method classify with morphological information?
Absolutely. So here in the demo, you saw me use just marker intensity. I did showcase just a little bit of that where you can change that from the standard to the custom. And in the custom, you can specify the exact measurements you want to use. Some of the morphological measurements are already calculated into Aivia, such as area, circularity, things like that. You can build a classifier just on those morphological measurements. You can build a classifier with the morphological measurements and maybe one or two intensity measurements. The combination that makes the most sense to you is really up for you to drive. So however you feel like you need to explore your data and to explore your cells, all of those things are possible.
Fantastic. All right. Well, that will conclude our session for today. So I want to thank you again, Dr. Tran, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast will be available to view on demand on November 15th, and LabRoots will alert you via email when it's available. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.